Hi, everyone. Thanks for joining us today. I'm Katie Goldsmith, one of the co-coordinators for MACAN. We're just going to wait another minute or two here for some additional folks to log in, and then we'll get going. Thanks for being here. Okay, well, we're a couple minutes past the hour, and I think a lot of folks have been able to jump on. So why don't we get going here? Hi again, I'm Katie Goldsmith. I'm the program manager at the Mid-Atlantic Regional Council on the Ocean, or MARCO, and I'm also the co-coordinator for MACAN, along with my colleague Grace Saba, who is also on the line. Today, we're excited to have Janet Nye present on her acidification monitoring work in the New York Bight. But before I get into that, as most folks know, this is the first webinar in our third series. We had a few scheduling changes for the remaining webinars in light of the expressed interest from MACAN members to stagger these webinars a little bit more. And so I just wanted to remind everyone that our next webinar will be February 13th on the Ocean Acidification Data Stewardship Project. On March 5th, we'll have a presentation about monitoring using pH gliders. And on April 2nd, we will have the Delaware River Estuary Nutrients and Coastal Acidification presentation. As usual, as in the past, all webinars will be recorded and they'll be posted on the MACAN website at the link you see at the bottom of this screen. So back to today, as I said, Janet Nye will be presenting on her team's efforts to date in monitoring in the New York Bight. Dr. Nye is an associate professor at Stony Brook University's School of Marine and Atmospheric Science. She is a quantitative fisheries ecologist interested in the effects of climate variability, climate change including ocean acidification on marine fishes and ecosystems. Her research has focused on how changes in the physical environment scale from individual physiological effects to populations and ecosystems. She's an expert in ecosystem-based fisheries management and teaches courses in fish ecology, biological oceanography, and science decision support. We'll have about 25 minutes at the end of the presentation for any questions and comments, and all participants will be muted during the presentation, so please ask your questions using the question box via your control panel on your screen. Anthony Himes, who's a PhD student at the Virginia Institute of Marine Science and the Virginia Sea Grant Fellow who's been assisting with MACAN efforts as of late, will be collating those questions for us at the end of the presentation. So feel free to submit your questions at any time during the presentation and he'll collate them for us. Final reminder, as I mentioned before, these webinars are recorded, so today's webinar is no different. We will be recording it and putting it on the MACAN website as well. Without further ado, I will pass the controls over to Janet.
Okay, thank you, Katie. Can Can you see my title slide?
Yep, you're good to go.
Okay, great. Well, thank you for that introduction, and thank you, Katie and Grace, for inviting me to talk about my research and our new ecosystem monitoring project for the New York Bight. As Katie mentioned, I'm a fisheries ecologist, and so the way that I've kind of gotten into ocean acidification research and monitoring is really trying to understand the effects of climate change and all the various stressors that come with climate change, including temperature, large-scale ocean circulation, and acidification, how those combined affect organisms directly and indirectly to affect fisheries production and ultimately ecosystem stability and resilience. So as you all know, OA is generally bad for marine organisms, and I really like this meta-analysis that done by Croker et al. In 2013 that combined a lot of different laboratory studies on many different marine taxa, and they looked at different endpoints, pooling all these taxa together. And in general, what you can see is that the mean effect size is negative, meaning that the effects are negative as a result of OA for survivorship, calcification. So we have lower survivorship, lower calcification, lower growth, slower development, decreases in abundance in most marine organisms. Little to no, at least statistically significant effect on photosynthesis and metabolism. But these are all based on laboratory studies. And when you separate these out by taxa, you know, the results are a little bit more nuanced. And so obviously shell-forming organisms are the most sensitive. And but when we look at the sort of taxa that I'm typically interested in, crustaceans and fish, what we notice is that you know, you get variable results, or there have been many many species that we haven't really looked at this in, or not enough studies to really make some conclusions about. And then when you look at primary producers, there's some evidence that, you know, OA can be positive. And then, you know, lastly, I would say that we don't fully understand how ocean acidification might affect predator-prey interactions and ultimately ecological processes. So, you know, even though these laboratories are critical to understand the sensitivity of different organisms to ocean acidification, the indirect effects of ocean acidification may be very severe. And so ecosystem models have shown, you know, when you have reduced survivorship and reduced abundance of your calcifying organisms, that affects those upper trophic level organisms. And so this sort of motivates my past work. So I'll just take 2 slides to talk about some of the work that we've done to try to take laboratory results and scale them up to population level and ecosystem level processes. So this is some work that is in review right now, led by Jason Greer at the US EPA, where we've taken the extensive laboratory results from Chris Gobler's lab here at Stony Brook for hard clams and bay scallops, come up with a response curve to different levels of PCO2 and, you know, basically a survivorship curve for them under different levels of PCO2. And we've then also developed matrix population models using field data, combined laboratory and field data to look at the population level consequences of these sensitivities to PCO2. And so, as you can see from the laboratory results, bay scallops here, A. irradians, is more sensitive to ocean acidification. And that's reflected here in the probability of a 50% decline of the population. But you can see that, you know, hard clams are still very sensitive to ocean acidification, or a doubling of ambient PCO2. And so this is an approach that we could take for lots of different organisms to scale these laboratory results to population level results. And then my student, Teresa Schwimmer, who, like Anthony, is a NOAA Sea Grant Mid-Atlantic Ocean Acidification Fellow, is looking at mechanisms by which larval and early life stages fish can cope and adapt to climate change. So we're working with Hannes Baumann at the University of Connecticut to look at the mechanisms that these larval fish, these are larval 1 day post-hatch, 1 DPH fish, Atlantic silversides, that have been exposed to different levels of PCO2 and at different temperatures. And what we're looking at is the ionocytes. So all these little purple dots here are ionocytes, or probably what you've learned is chloride cells. When they were first discovered, they were discovered for their role in regulating the salt balance in fishes. But in addition to being able to regulate chloride and transport chloride either in or out of the cell, depending on if you're in the fish is in freshwater or saltwater, these ionocytes also regulate pH. So this is probably the mechanism by which fishes can really be relatively insensitive to ocean acidification or changes in pH. However, as the larval fish goes through this ontogeny, this great period of change from embryos to larval fish or juvenile fish with gills. These 1 day post-hatch fish have all the ionocytes on their skin, but as they develop, they get ionocytes in their gills, and that could be a really critical area that explains some of the lower survivorship and growth that we see in Atlantic silversides with this work. So, but what Teresa has been doing is looking at the ionocytes as well as metabolic rates, and in 1 day post-hatch larval silversides, we haven't seen very little effect of PCO2 on metabolism. There's very little effect on the number of ionocytes. But in work that she's done with a high school student this summer, it does appear that instead of changing the number of ionocytes, they may be upregulating different genes that help regulate acid-base balance, and downregulating some genes that affect disease resistance. So we're doing a battery of these studies looking at different endpoints in fishes and early life stages fishes, and then Teresa's goal, like much of the work in my lab, is to translate that into population and ecosystem processes. So you can see in my lab, we're focusing on 2 aspects of the vulnerability of organisms to climate change and ocean acidification. So the vulnerability of an organism is a combination of its sensitivity in this pink circle to, let's say, low pH, its ability to adapt to that low pH. So in fishes, that's sort of the ionocyte piece that we're thinking out. But an organism can be sensitive and have low adaptive capacity, but never be really exposed to those stressors. So like the third component of vulnerability is its exposure. So the most vulnerable organisms are going to be those that have all 3 of those things, that are sensitive, have low adaptive capacity, and are exposed to those conditions. And so that's really where my interest in monitoring carbonate chemistry comes into play, and why I got into this for the offshore monitoring project. So the New York State has been great to work with. The Department of Environmental Conservation, or DEC, has been great to work with. And since I've been here about 7 years, they've been working on this New York State Ocean Action Plan and released it in 2017. I encourage you to take a look at it. It's really ambitious. There's 61 action plans, and within each one of those 61 action plans, there is actually a roadmap to meet those goals and implement those strategies. The ultimate goal being to implement ecosystem-based management for the New York Bight. And of those 61 action plans, we're involved in quite a few. Obviously, one to monitor ocean acidification and then investigate impacts of ocean acidification on shellfish and crustaceans, and to establish a baseline ocean monitoring system for the New York Bight and develop ocean indicators that monitor ecosystem health for the New York Bight. So this was released in 2017, and then almost at the same time, I did want to mention, if you do take a look, as part of the Ocean Action Plan, they really solicited the opinions of the public and scientists, the variety of stakeholders in New York. And this is just one example of some of the research needs that were identified for New York State ocean ecosystem from our New York Marine Science Consortium. And number one in those was design and sustain a long-term integrated monitoring program compatible with regional federal efforts. So that's what this ocean acidification carbonate chemistry monitoring is coming from. Number three is looking at water quality indicators, including ocean acidification. But sort of simultaneously with this development and implementation of the Ocean Action Plan, the New York State legislation and Governor Cuomo signed into law the New York State Ocean Acidification Task Force. And again, there's a lot of sub-goals, but the two main goals of the OA task force in New York is to identify the causes and factors contributing to ocean acidification and to evaluate the ways of addressing the problem by using the best available science to look at ocean acidification and its impacts. And so this is these are some pictures from our first meeting. This is the Ocean Acidification Task Force itself on the left. And I'm sort of showing you the opposite view where we have we had really good attendance from the public who are interested in this issue. Now, this is the New York Bight, the area that New York State and DEC is primarily interested in, which basically, if you draw a line from the southern part and the northern part of Long Island, where we are at Stony Brook, they want to develop have wanted to develop an offshore monitoring program for this region. And so we worked closely with them to design a monitoring program and are still working closely with them to monitor a lot of different things, as many different things as we can, as is possible. And so before doing that, what we did, as well as part of a MACAN working group that looked at monitoring, was to really assess the needs for monitoring in the New York Bight. And so I want to give a shout out to the Marco web portal, data portal, for allowing us to just really look at the data needs very quickly in New York Bight. So you can see this area, New York Bight, is really lacking in carbonate chemistry measurements. So these are showing you some of the fixed station and continuous monitoring sites that are mostly in estuaries and coastal areas. For the New York Bight area, we have cruises. We have to use cruises to get carbonate chemistry from this area at this point. But you can see a lot of those cruises have missed the New York Bight area. And if you break that down into the number of parameters that are taken, as we know, most of the monitoring sites are only monitoring one parameter, usually pH, supplemented by periodic water sampling. But on these cruises, we typically have two parameters, and very rarely we have some some cruises that have done 4 parameters as shown in red. I believe those are the ECOA cruises. And if you break that down by the different carbonate chemistry parameters, you can see that pH and PCO2 are the 2 parameters that are most frequently measured, and that makes sense given that those are the most easily or readily available, commercially available equipment that you can get, while DIC and TA is a little bit more expensive and hard to do. And so, but again, the point being that New York Bight is really missing a lot of measurements. And these measurements that I'm showing you here, again, are not frequent. These are conducted by cruises that happen, every other year at the most. The best information that we have about the New York Bight is from the ECOA cruises. This is a paper that was from the 2012 ECOA cruise that we looked at a lot in designing the survey. This figure is showing you the surface aragonite concentrations, and so you can see north of Cape Hatteras that aragonite concentration is lower than it is, sort of south of Cape Hatteras, and then these dots are showing you the transect. So, all the maps that I showed you before are primarily surface measurements of carbon dioxide. We have very few measurements of bottom carbonate chemistry or carbonate chemistry at depth, and that's I'll show you in a bit why that's important. If we look at cross sections up and down the coast from the ECOA cruise, again you can see this is Louisiana, Florida, Georgia, and once you get above Cape Hatteras, what you see from this New Jersey transect is a low aragonite concentration area just above the bottom of the surface, and that's basically the Mid-Atlantic cold pool, or the remnant winter water that is very cold, very important for lots of more cold water benthic species, but it also traps a lot of CO2, and that's right at the bottom close to where our very important shellfish are. And you can see this also low aragonite areas in the Gulf of Maine and New Hampshire, in the bottom waters there, also close to where our shellfish would be, and exposing them to potentially poor conditions. Basically this one transect is what we knew when we sort of designed this program, is and we knew we wanted to get and quantify the spatial and temporal extent of this cold pool, low aragonite area, and come up with indicators as part of the project of the volume and extent and perhaps duration of this this area. So what we have embarked upon is to do seasonal cruises, so 4 cruises a year for the next 10 years, and we will try and develop indicators to get the spatial and temporal extent of this and look at the seasonal dynamics of that area. We're using our research vessel, the RV Seawolf, as shown here. This is actually a picture taken by a drone on our cruise, and as you can see, the Seawolf is a transformed fishing vessel. It used to be a fishing vessel that was transformed into our oceanographic research vessel. It's primarily been used in the past for fish trawl surveys. So we've added quite a bit of equipment to the boat to be able to do this work. We have done 1 cruise so far, so we plan to do seasonal cruises. We've done 1 cruise this past July. That was our first cruise, and this is the cruise track that I'm showing you here. So we start in Port Jefferson in the middle of Long Island, and then we move out offshore So we get part of Long Island Sound, and then we have about 8 transects that we don't expect to do every cruise period, but we did almost all of them in our July cruise because we had absolutely perfect weather. So it was an amazing cruise in terms of weather. We got a lot done. And so what we've installed on the Seawolf is a new seawater system that has this underway system. So we have a General Oceanics PCO2 system. We have a thermosalinograph. And so we're also measuring fluorescence, CDOM, and turbidity, and that's what we measured on our first cruise. And since then, we've added a sunburst pH underway system, and a DO probe. And of course, we're doing CTD casts periodically on our cruise. But this is not just a carbonate chemistry cruise. We're also, the entire time that we're underway in measuring PCO2 and pH, we're measuring fisheries acoustics. So this is some data that was generated from our first cruise by Joe Warren. And he tells me these blobs here are fish, but we can basically get from this, the volume and abundance of different fishes as well as zooplankton. So at the same time we're getting carbonate chemistry data, we can look at lower trophic levels, fish and zooplankton. We're collecting periodic samples of zooplankton, and this is showing you sort of the biovolume, some of our preliminary data, and some of the species that we're catching with that. And I think uniquely during this cruise, during the day, our effort is focused on marine mammal observations. So we're actually, we have observers of marine mammals quantifying and identifying different species. And so these are all the different species of marine mammals that we have identified, and again, that's going to be combined with all this data from multiple trophic levels, and then also the physical and chemical data. So then getting into some of the actual results that you're probably interested in, the PCO2 measurements. Again, this is our cruise tracks shown in blue at the bottom. And then this is our sea surface salinity, and I just wanted to show that we're sampling basically 3 different regimes in the New York area. So we have the lower salinity area, the estuarine waters of the Long Island Sound, and the, sort of the East River, or Hudson River, and East River too. But and then the high salinity, more oceanic waters shown in red. And then the yellow areas, as you'll see, is lower salinity water influenced by the Hudson River plume. And then these are the numbers from our underway PCO2 sensor. This is fugacity of PCO2, so you can see the higher numbers in the estuarine waters, lower numbers here in the more coastal waters. And if we plot the fugacity against sea surface temperature, you can really see the different dynamics that are ongoing in these different regimes, where you've got these higher CO2 values, and then lower in the more coastal waters. These are our CTD values shown at the surface. So I'm showing you these data maybe against my better judgment, but I do want you to note that for the pH values, our pH numbers are most likely off. They're definitely off. And so we realized that the pH sensor was out of calibration, and then when we went to calibrate it, it broke. So we're going to have to rely on the water samples that we've taken during the CTD cast. So I'm including the pH just so you can see that the patterns are the same, but really when we to look at the absolute values, we're going to have to use the water samples. But these are the CTD surface values. You can see, again, the different regimes that we're looking at. So if you look at salinity in particular, you can see that lower salinity water, the Hudson River plume, and then the more oceanic water. And if you, as you go below the surface, what you start to see is, again, that plume, but also some lower areas of lower dissolved oxygen and lower salinity, or sorry, pH. Yeah, so particularly here at 25 meters, you can see this lower dissolved oxygen and lower pH. And as you move further out, we're getting into deeper water, into this coastal water. Now, then what has really been interesting to me is to, again, look at the cross sections of the shelf. So this is showing sort of one of our more eastern cross sections, and now you can clearly see the cold pool if you look at the temperature as well, and that is also, where we see our lower pH and lower oxygen values. And then as we move further to the west towards New York City and the Hudson River plume, you'll see that as well. This is sort of the middle of Long Island Sound, the cold pool, and lower pH values there that persists. And then you see that as we go towards the Hudson River plume. And so we're working on methods actually to kind of take these transects and cross sections and use some quantitative techniques to, not just contour, but be able to predict and be able to develop indicators that give us, again, the duration, extent of the cold pool and these lower oxygen and lower pH areas that will expose organisms to potentially negative environmental conditions. Okay, yeah, that's what So these are that's kind of showing that, putting those cross sections together to get an idea of spatial extent and duration if we do this over seasonal timescales. So I want to bring you back to this this sort of conceptual framework that we really need to look at sensitivity, adaptive capacity, and exposure. So I think this is a really unique opportunity to be able to look at the seasonal and temporal variability in carbonate chemistry, as we're able to do this very relatively intense spatial analysis of carbonate chemistry seasonally, and then over a 10-year time period, and hopefully longer. So in conclusion, I kind of wanted to emphasize that really to understand the effects of climate change and ocean acidification on ecosystems and organisms, we have to focus on these three components that affect vulnerability, because really, and we've been working closely with New York DEC and stakeholders, they know intuitively that those, we need to understand all those three things to really take action, against climate change and other, to do mitigation and adaptive strategies. And to, my interest is basically to assess the effects of climate change, not just one stressor, but multiple stressors on organisms and ecosystems. So to do that, you have to consider simultaneously many stressors impacting marine organisms. And certainly the experimental work that's ongoing has been moving in that direction for many years now. But also we need to look at both the direct and indirect. So direct in terms of the sensitivity of organisms to ocean acidification, but indirect in terms of trophic interactions. And so I would sort of challenge our community to implement more studies like this that are multidisciplinary, multi-trophic level, and to kind of think of these things at the same time, as difficult as it is. I think this New York State offshore monitoring project has been really bold in trying to do this really challenging thing. So I want to give a huge shout out to the New York DEC for funding this offshore monitoring project. It's going to be really invaluable to us and to the community. I also want to thank NOAA and NSF that have funded my other work in ocean acidification. I have to recognize the other PIs on the project, Leslie Thorne, Joe Warren, and Charlie Flagg, who are all at Stony Brook with me. Our wonderful eShop people who many of you may know, Tom Wilson and Alex Nedden. My postdoc, Sean, has actually developed, she actually put a lot of these figures together, so I should give a huge shout out for her, and then all the other students and techs that have gone on the cruise. It's they have to be multidisciplinary, so they all know how to do each of these jobs. They have to be chemists and biologists and marine mammalogists at the same time, and of course our boat crew. And as we kind of organize to get the questions ready, I'll just give you a little bit of eye candy. This is from, it's really rewarding, as I'm the carbonate chemistry expert on the cruise, but we also get to see some really cool stuff out there. So I will take your questions.
Thanks so much, Janet. That was really great. I will turn it over now to Anthony Himes, as I mentioned, who's been helping out Maycan as a Virginia Sea Grant Fellow to collate those questions and send them your way. Anthony, go ahead.
Great. Thank you, Katie. So we do have a few questions here. The first one, I guess we'll start with, and this ties through with some of the other ones, but initially, Janet, you showed some data for, or showed some pictures of silversides, and we had a question, as you talked about kind of the developmental timeline and their susceptibility to OA, do you have an idea, or does the community have an idea of how much that varies across different species, and if some species would then be more vulnerable than others?
Yeah, that's, well, so for a species like silversides, they are an estuarine fish, and they experience a wide range of salinities during their life history. And so it's thought, because they do have to adapt to that they are less sensitive to pH changes. Because not in addition to salinity changing, pH changes quite a bit in the estuary as compared to something like the sand lance, which Hannes Baumann, the co-PI or main PI actually on that project, has found is one of the most sensitive fishes to acidification. So the general idea, and I think the consensus among the community right now, is that those species like Atlantic silversides that are found in estuaries should be less sensitive, or at least have more adaptive capacity than something like the sand lance, which has not evolved to live in an environment where pH and salinity doesn't change very much. So again, you have to weigh the sensitivity and the exposure of those two organisms to ocean acidification. And sort of related to that question is, some fishes have more direct development where there's a shorter time basically between when they develop ionocytes and gills and things like that. But there's others that may have a very long, time where they don't have those things that enable them to be to cope with acidification, or they may have like a different sort of transition to between those stages that make them more vulnerable.
Okay, great. Thank you. So the next question we got was, how do you foresee the data you collect in these monitoring efforts being used? And then there's some suggestions like in modeling or to influence the design of lab studies that consider multiple stressors or other uses as they relate to the New York Ocean Action Plan.
Oh gosh, okay. So where to begin with that? So one, as wonderful as I think this is going to be one monitoring program where we have pretty high spatial and temporal resolution, but it's still not complete, and we're still going to have to use quantitative methods to contour and get a full picture of what the extent of the environmental parameters are. So I think the first thing that we're going to do, and my postdoc John is sort of charged to do, is to develop methods where we can go even further and try to extrapolate between our sampling times, and using things like neural networks, as well as connecting these to like ROMS models, physical models, to look at the dynamics. I think just being able to describe the seasonal cycle in carbonate chemistry is second on the list, first or second on the list actually. And then really part of this offshore monitoring project is to start doing the monitoring, but we're going to constantly be evaluating how can we leverage different other people that are sampling similar things, so we don't duplicate effort, and where someone might be monitoring something else perfectly well, we can use that data, but then we can move our resources and time into sampling something different, so that we really get a holistic view of the system. So we do expect that the monitoring plan will change, and so in terms of the monitoring, that is definitely we're going to optimize that essentially using quantitative techniques. And then in terms of informing experiments, obviously the big application is looking at sea scallops, surf clams, and things like that. I think we're still lacking on data taken at below the surface, at depth. So we're hoping to work with Grace on using gliders. Charlie Flagg actually has a glider as part of this offshore monitoring project, in which we're going to get hopefully a pH sensor, and then we'll have more data on the bottom conditions. But what we should be able to do is, again, get the duration and extent that the organisms are exposed to, and so you can use some of that great laboratory data to scale up to what should be the population level impact, or what should be the total survivorship of these organisms.
Okay, we have a few more questions coming in. Kind of going off what you just kind of talked about, the gliders, we had a question about, would a fixed array of sensors on the shelf fill in the gaps that are missed by the cruises?
Yeah, we thought about that, but it's very expensive to have a fixed array. You still need ship time to go out there and maintain those sensors. So the gliders seems like a more effective option. Joe Warren, who's the acoustician on this project, is going to be putting bottom acoustic sensors that look up and get acoustic data on different parts of the ecosystem. And so we've talked about maybe putting pH sensors on those, so then you would get that data too. And there's other efforts by people at Stony Brook and elsewhere, like that are doing are tagging fish, and so they have acoustic receivers on the bottom, and so we could attach pH sensors to those, and that would help get us some pH data for those bottom things. And then we've also, it would be great to have like some of those sail drones with pH probes on them, but I'm racking up a pretty large bill right now. And the other thing too that gives me pause is that some of those things we're not quite there in terms of technology, where it's easy to get a CFED or some sort of pH sensor, but you really want to have two parameters. So I think that in a few years we'll be able to put maybe two of those things together at an affordable price and maybe get better coverage in combination with our shipboard measurements.
Okay, kind of going off of that, our next question was, at 4 cruises a year, do you think you can capture most of the variability in parameters that drive OA in order to make better decisions?
Well, I guess we'll find out, right? I think so. I think there's some additional information that might be good to have. I think we're going to have to get data, like satellite data on, when the timing of the bloom occurs. I think we're going to have to use that. We're working also to look at nutrients and chlorophyll to try to get at those drivers, but that's not fully implemented into our sampling protocol yet, just because we're doing so much already. But I think combined with physical models, we have a really good ROMS model. I think combined with that physical model and what we know about, the chemistry and physical dynamics, I think we can get at a lot of the dynamics. Can we get at it in a way that's meaningful to stakeholders and decision makers? That's what we really have to see.
Okay, we had a question asking if you could speak to if the chemical analyses that were done on your cruises were in line with the expectations from before the cruises began.
The expectations. I would say we've only done one cruise, and we're calling this our Shakedown cruise. And I thought it went amazingly well, partly because our weather was just perfect. We're going out in February, so that is, I think, going to be the true test. And we also have all of our equipment, on the boat. So last time we only had the underway PCO2 sensor that, was up and running and collecting data on July 13th, and we left it on July 14th. So it went amazingly well. So I'd say it definitely met our expectations. And now we just have added equipment, and I think it's going to be better. I think we have a lot of water we have to sample now. So I was only really able to show you PCO2 and pH, but we have a ton of water samples where we'll actually be able to get to aragonite and the other parameters. And so we'll see how, what our variability and our error is with those things.
Okay, kind of. I think this is in regards to the kind of duration and persistence of the cold pool, but how many seasonal monitoring events are planned in your project?
So we have 4 7-day cruises planned, where we're going to go from Long Island all the way out to the shelf. That's our offshore monitoring project. But the PCO2 sensor is going to be on the boat the entire time. So we hopefully will have be able to collect some sort of opportunistic samples on the other cruises that the Seawolf takes and the CTD casts that they take on other cruises. So I think still, those bottom temperature measurements are the ones that are hardest to get, but we'll definitely get very well the seasonal cycle in the surface waters, and especially closer to shore. So, we have a ton of other surveys that don't go all the way out to the shelf, but are sort of closer to Long Island that we'll be able to get some really good seasonal information about.
Okay, I think this is our last one, unless any more come in. But it's once the data is collected, will it be made available somewhere online, or is there a plan for if people might be able to access the data?
Yeah, so because this is funded by the New York DEC, it basically Stony Brook has rights to it for 3 years, and then it goes to the DEC. The DEC definitely wants people to use it. So, if you're interested in collaborating with us, just get in touch with me and we'd be happy to collaborate.
All right. Thank you, Janet. I think that's everything I have here, unless anything came through to you, Katie, and then I'll turn it back over to you.
All right. Thanks so much, Anthony. And thanks again, Janet. That was really great. Thanks for everyone for joining. Like I said, we will have this posted on the Maycan website to spread and share the word as soon as we can. And thanks again. Have a great rest of your day. Bye, everyone.